Thanks for the invitation to present here today. I'm really excited to be with you here. I'm going to review some of the data you've probably seen on why we're using immunotherapies for lung cancer and other malignancies, but I hope the rest of the talk will challenge the way we're currently thinking about PD-L1 and PD-1. So here are my disclosures. So, like I said, the objectives are really to recognize how we're using immunotherapies in thoracic malignancies, but to also think about predictive biomarkers for response to immunotherapy, and also to communicate some of the challenges using the current biomarker strategies. So this wasn't too long ago, but when I was a fellow, this was our approach to metastatic disease. We had chemotherapy, chemo. We had one EGFR drug approved, and if that failed, we then went to more chemotherapy. But the last year and a half has really revolutionized what we're doing, at least for stage 4 disease. And in 2015, Opdivo or nivolumab was approved for lung cancer and subsequent lines of therapy. The day that was approved, the email came through. Our melanoma colleagues already had this on the shelf, and we started writing for that an hour later. And we've seen some phenomenal activity. We'll cover that in a little more detail. But then atezolizumab was more recently approved, and then pembrolizumab also. So we have 3 drugs currently on market and many more in development, and it's a little confusing as to which drug you use or in which setting. So with these approvals and with some others, we're not going to cover targeted therapy so well, but we now have immunotherapy established in the front line of non-small cell lung cancer, in the second line, and we also have many more targeted therapy options, but we're just going to skip those for this talk. So here are the 3 drugs that are approved. I just listed them in order of their approval. Nivo, Pembro, and Atezo all have second-line approvals, but only pembrolizumab is approved in the front line, and I'll show you the overall survival data as to why that is. Each of these drugs was developed with a separate companion diagnostic test, so they each have a different antibody to detect PD-L1, and they recognize different parts of PD-L1. And these drugs and the others in development use different immunohistochemical cutoffs for what's considered positive or negative. So for Nivo and Atezo, they look at 1% or greater. For pembrolizumab, our current approval is for patients with 50% or greater expression. Durvalumab has a 20-25% cutoff, and the other drugs don't have their cutoffs established yet. But there are differences with how these drugs are given, where nivolumab currently is on a 2-week regimen, whereas Pembro and Atezo are every 3 weeks. So we have differences with how frequently these are administered and what sort of companion diagnostic testing is required, and that's sort of influenced how we're practicing. And I'll explain that as we go through some of the data. But this slide's a year and a half old now, but it's still very meaningful. And what this did is summarize all the PD-1 and PD-L1 trials that have been published as of a year and a half ago. And what you see are the response rates based on all comers, PD-L1 positive patients, and PD-L1 negative patients. Those who had positive expression across different tumor types roughly have a 50% response rate with immunotherapies, whereas those who are negative still have a response rate, and that's closer to 15%. And unfortunately for a lot of our cancers, that's better than our old standards of care for second or third line therapies. When you look at all comers, that averages out to around 30%. So first of all, do we really need a test for PD-L1 when these drugs work better than docetaxel, gemcitabine, vinorelbine, and those other agents? So we'll go through the data for nivolumab in a little more detail since this was the one that was first approved. And this is the progression-free survival. And this actually doesn't look that impressive. You see the curves overlap in multiple parts. But at 1 year, you see that nivolumab has more than double the progression-free survival than docetaxel. And you also see this little tail towards the end at 2 years where there are patients who still have not progressed on therapy. It's only a small number, but that tail is there, and the overall survival data are more clear. But this is not statistically significantly different. The progression-free survival is not significantly different between nivolumab and docetaxel. But the overall survival data are, and here you see I think this is a 12% difference in overall survival at 1 year. This was significantly different, but the figure that really shows how these drugs are different is the swimmer's plot. I don't know if you look at these as much as we do in medical oncology, but they took all the responders. This was a randomized trial. They took half the patients, gave them nivolumab, the other half got docetaxel, and these are all the patients who had a response. And then what we see here, every bar represents the duration of all the documented responses. So first of all, there are many more blue bars than green bars. So many more patients responded with nivolumab than docetaxel. And those responses also lasted a lot longer with immunotherapy than they did for chemotherapy. For both drugs, most of the responses were found with the first response assessment around 2 months. But there are some cases where you'll see these black dots where the first response was documented that are almost a year into therapy. That's not as common, but that can happen. And where you see red, that means the drug was stopped, but the response continued. So you see a little bit of red here that even though the drug is stopped for toxicity, some patients still benefit beyond that. So overall, there just seems to be, when patients benefit from immunotherapy, it's longer than what we see with chemotherapy, and more of them benefit. Where it starts to get interesting is how you select which patients to give the drugs to. This trial was more of an all-comers trial, regardless of PD-L1 expression, and what we're seeing here are the the survival data based on different cutoffs of PD-L1. And here BMS used greater than 1%, 5%, and 10%. And you see the curves seem to separate a little bit better with each cutoff, but not terribly differently. So despite it being maybe a little more enriched in the higher PD-L1 expressors, this drug got approved for all comers because regardless, it was better than docetaxel. The data for pembrolizumab and atezolizumab are fairly similar. Once you've seen one of these second, third line trials, it's fairly similar to the other drugs, so I'm not going to cover that. But where we have different data is in the front line. And this was just presented towards the end of last year. And both pembrolizumab and nivolumab were compared to standard platinum doublet therapy in the front line. They used different PD-L1 cutoffs. And what we saw was pembrolizumab was better than chemotherapy in the front line for non-small cell lung cancer. They used a 50% cutoff to select which patients should get the immunotherapy. We participated in this trial and we screened over 20 patients. Not one of ours met that cutoff, but they're telling me on average about 1/5, 1/4 of patients have greater than 50% of PD-L1. It wasn't our experience, but regardless, overall survival was significantly better. The response rate was around 40%, and when they responded, it was durable. Nivolumab did not, the trial did not include a 50% cutoff. They just required 1% or greater expression. And with them, they still haven't published it, so I had to take the image from Twitter, so I'm sorry. But on the right is the overall survival data, and you see there's no difference between nivolumab and chemotherapy. So currently, we only have approval for pembrolizumab with 50% or greater PD-L1 expression. So that's why we're checking it in all patients when they're coming in a clinic for frontline therapy, and we're trying to give them immunotherapy instead if they qualify. The data I'm going to show you later may challenge our use of PD-L1, but we're playing within the rules we have, and we do see a survival benefit with this drug. We really don't know why nivolumab failed and pembrolizumab was successful. Like I said, it hasn't been published. The second one hasn't been published, so we still don't know. There's now talk about whether one antibody is better than the other, but we still really don't know why one failed and the other didn't. When you look at these trials and how they assess PD-L1, up until these frontline trials, they have all used essentially archival tissue, and I'll show you data on the timing of PD-L1 expression. And looking at an archival specimen, I don't think is going to give you the best readout of PD-L1, so I'll explain that in more detail. They also allowed any size biopsy specimen, and we'll talk about data as to why that might not be appropriate. And very few studies looked at tumor-infiltrating lymphocytes and why those may or may not be important. So when we ask about PD-L1 expression, the first question is which antibody should you use? There are now 3 companion antibodies and many more research antibodies. This was before we had approval for the companion diagnostics, but here we're looking at 2 antibodies just comparing PD-L1 expression with one another. On the left, we have whole tissue sections, resected lung cancer. On the right, we have a TMA. And this was an immunofluorescence project here where we looked at, Dr. Brim's group at Yale looked at PD-L1 expression based on these specimens. And what you see on the left, there's about a 26% discordance in what was called positive or negative in whole tissue sections. Whereas on the right with that TMA, that number goes up to around 36%. So 1/4 to 1/3 of patients have discrepant PD-L1 expression just with these two antibodies alone. We have some data for other antibodies, but it's just really discouraging that 1/4 or more of our patients have discrepant readouts based on what antibody you use.
What's TMA?
I'm sorry, tissue microarray.
Thank you.
So on the left is whole tissue sections, on the right just real small specimens. So the smaller the specimen, the more the discordance. Sorry, it's not advancing. There we go. So this was just published, I think, a month and a half ago now. This is called the Blueprint Project that Fred Hirsch from Colorado is running. He took the 3 companion antibodies and the one that's expected to be available next, and here they looked at 40 specimens for PD-L1 expression, and every column represents a different specimen. And 3 of this antibodies seem to correlate relatively well. They didn't provide any statistics, it's just a very descriptive report they gave. But one of the antibodies, and this is the companion diagnostic for atezolizumab, and that's in that teal blue color, consistently underscored PD-L1 compared to the other 3 antibodies. So as we're trying to sort out, can you use one antibody to determine PD-L1 expression for these drugs, it seems like one of these is consistently detecting it by 10, 20 or more percent. So if you're using a 20 or 50% cutoff, that degree of variance is really significant in determining who's positive for PD-L1. And one of the reasons for this might be is the epitope recognition of each antibody. The 28A and 22C3, these are the nivolumab and pembrolizumab companion antibodies. These both recognize extracellular epitopes of PD-L1, whereas that Spring Harbor 142, the one that underscored PD-L1 compared to the others, it recognizes an intracellular epitope. So we've known for years that PD-L1 can be shed from the surface. So this just begs the question as to what's happening physiologically with the expression and why these are not detecting it at the same rate. So this is some surgical data from France where a series of, I think it was 140 specimens that had a pre-surgical biopsy and a fully resected tumor were stained for PD-L1. And they looked at the detection of PD-L1 between the biopsy specimen and then the fully resected tumor. And what you're seeing here is about 3/4 of the biopsy specimens were determined to be negative. There's no PD-L1 expression on the tumor cells or the immune cells. But once they looked at the fully resected tumor, about 50% of those turned positive that were negative. So if you're just looking at a small FNA or a small core needle biopsy, these data suggest that 50% of your patients may have detectable PD-L1 that did not in the biopsy. And that's just really discouraging when I'm trying to figure out who do I give these drugs to or not, when 50% of the time our biopsies might not show us the full picture. So we've been really interested in the heterogeneity of PD-L1 expression, but we haven't been looking at biopsies compared to fully resected tumors. We've been looking in both multifocal lung cancer and paired primary and metastatic lesions for how PD-L1 and tumor-infiltrating lymphocytes correlate between these paired specimens. So here we were able to put together a series of, I think it was 73 patients who had a resected lung cancer and later had oligometastatic brain metastases and had that resected by our neurosurgeons. And we looked at both PD-L1 and T cells between those pairs. And here's just one case where we see PD-L1 was prevalent in both specimens, but there were almost no T cells in the brain metastasis. And here's another case where PD-L1 and T cells were both missing from the brain metastasis, but were in the primary. So when we looked at that across all of our pairs, Statisticians prefer, this is called a Bland-Altman plot or limits of agreement. They prefer this to correlations. It's essentially the difference between the pairs plotted over the average. And what we're seeing is in some pairs we had almost 100% expression in one lesion, but 0 in the other. So there are many cases where we have 20 or greater percent difference in PD-L1 expression between these pairs. And when we're using 20 and 50% cutoffs, this gets to be very significant. But we're trying to think about PD-L1 a little differently now than just this binary marker as it's present or not, and you should give Pembro or another drug or not. And I really think its expression is contextual and needs to be considered that way. And here's one of the first paradigms as to how to think about it. And the first way to just take it beyond this binary, is it there or not, is to look at the presence of an immune infiltrate or not. And it's hard for me to read from here, so I'm sorry if it is for you too, but here it's just breaking it down based on PD-L1 and tumor-infiltrating lymphocytes. And on the left is where you have the presence of both, and we're calling these adaptive. And this is where it's thought if you block PD-L1 or PD-1, you have the presence of an adaptive immune response, and that is where you're going to get the attack of the tumor. Whereas on the right, that one is immunologic ignorance, where you have no PD-L1 expression, no tumor-infiltrating lymphocytes. So giving PD-1 or PD-L1 inhibitors to these patients may not be the best idea. Then on the bottom right, I believe that one's intrinsic, where you have PD-L1 expression, but you have no tumor-infiltrating lymphocytes. And we know that some different oncogenic molecular drivers can upregulate PD-L1, but its expression might be meaningless if you don't have an adaptive immune response attacking the tumor. So even if you detect PD-L1 expression and give one of these inhibitors, if you don't have an adaptive immune response, what's the point? And then on the left, bottom left, these are called tolerant tumors. This is where you don't have PD-L1, but you do see an immune infiltrate. And there are many other immune checkpoints that we're not going to talk about a whole lot today, but these patients may benefit from other immune checkpoint blockers than PD-1, PD-L1 inhibitors. So we took our series and we just categorized our specimens as to whether they were adaptive, intrinsic, tolerant, or ignorant, and how that changed in the paired brain metastasis. So this is a Circos diagram, and half our reviewers loved this, the other half hated it. So if you're in that latter category, I'm sorry. And the other thing is, if you make Circos diagrams and you send them to your co-authors, autocorrect changes it to circus, so just be careful. So on the left, you're just looking at the primary lung cancers and how they were categorized by whether they had PD-L1 and tumor-infiltrating lymphocytes. And the ribbons that connect them show you the changes between the pairs. So when you look at the top left, the adaptive, this is the group that has both PD-L1 and tumor-infiltrating lymphocytes. More than half of those lesions lost the tumor-infiltrating lymphocytes or PD-L1 expression or both. Those that were tolerant and had immune cells within the lung primary lost those immune cells in the brain metastasis. Those that were intrinsic and expressed PD-L1 but had some immune cells often lost those PD-L1 expression. It was very rare that we saw a tumor without PD-L1 or TILs gain that in the brain metastasis, although there are a few cases where that did happen. So this is the first time I'm showing this data. I made the point that I think the presence of an adaptive immune response to a tumor is really important, and we've never really characterized what is that response. So we've taken this same data set, and now with sequencing, we can molecularly characterize T cells, and we could determine how many different T cell clones are in each lesion and how abundant each of those are. So here on the left, we're looking at the number of unique T cell clones within the fully resected lung primaries. And on the right in blue, you're seeing how many unique T cell clones are in the brain metastases. And the purple is the overlap. And I think this is really fascinating. First of all, as the last Circos diagram showed, we're seeing many more unique T cell clones in the primary than we're seeing in a metastasis. And I thought that would be the case just from the IHC data we had, but I thought there would be much more overlap in the clones that we find in the brain than in the lung. But what you see here in purple is there's very little overlap between the clones we're detecting. I'm still trying to make sense of this, but we're seeing greater diversity of T cell response to primary lesions than the brain. Since we titled this talk Thoracic Malignancies, I thought we'd talk just a little bit about mesothelioma also. We looked at PD-L1 expression in mesothelioma, and roughly 40% of them have expression. This didn't seem to correlate with the tumor-infiltrating lymphocytes, but as we've seen in many tumors, this expression is correlated with worse survival. There are other studies where that's not the case, but overall, when you look at these meta-analyses, PD-L1 is a negative prognostic marker most of the time. Using similar specimens, but where we had pairs, where we had a biopsy and then a resection, or we had different sites of mesothelioma resected, we looked at the PD-L1 expression between those sites, and similar to what we saw in the lung data, this was 22% of patients had discrepant PD-L1 detection, and that's what we see in blue, with the primary lesions on the left and the secondary lesions on the right. 22% of them had differences in PD-L1 expression. So right now, immunotherapy is not approved for mesothelioma. There are at least 4 clinical trials ongoing exploring different cutoffs, but similar to what we've seen in other tumors, 25% discrepancy, I think, is a big deal to consider when you're selecting patients for treatment. The other thing about PD-L1, and this is hard to demonstrate in a patient, but its expression is dynamic. In the lab, we've known this for years. If you take tumor cells or other cells and you give them interferon gamma, PD-L1 is going to go up significantly. There are data where if you radiate a lesion, PD-L1 will go from low to high. We've taken here, these are 3 different mesothelioma cell lines with low, medium, and high PD-L1 expression. And in this case, I'm showing you, we just treated them with venetoclax, our common second-line therapy. And in all cases, we saw significant upregulation of PD-L1. So you can modulate PD-L1 expression, but if you biopsy a radiated lesion and PD-L1 goes up, does that really tell you that giving immunotherapy is going to be helpful or not? I don't really know, but I'm just trying to make the point that this is not a static marker. This is not like EGFR and ALK. It's not an exon 19 deletion or a rearrangement. This comes and goes. It's heterogeneous. The other thing about PD-L1, and this part's not talked about as much, but its expression is crucial to T cell survival. When the immune system sees a new antigen, it will expand and contract and leave you with T cells that most effectively recognize that antigen. And PD-L1 is critical to the survival of those T cells that survive the contraction phase. So that's what here we're just looking at PD-L1 expression in a mouse exposed to ova, and that expression goes up significantly during the expansion. And when we do more precise work, we see it's only those with expression that survive contraction. The other thing is when you knock out PD-L1 expression from T cells and you give them a tumor, give the mouse a tumor and give them these PD-L1 knockout T cells, they cannot effectively treat that tumor. So on the right here, and the far right of that bar, you're looking at mice who received tumors and then had PD-L1 knocked out T cells, and that did not control the growth of the tumor, whereas in the middle, we see the wild type T cells could. So PD-L1 is not just expressed by tumors, it has importance to immune cells, and that's why some of these antibodies, they're designed to remove parts of the TEL that promote antibody-dependent cytotoxic cellular cytotoxicity, so you may kill some of your immune cells with the wrong antibody. So the other thing we're doing, we're trying to sort out what is the best biomarker to select patients. And since if you biopsy one part of the tumor instead of another, that may not tell you if PD-L1 is expressed or if there is an adaptive immune response, we've been trying to define what is a tumor-reactive T cell. And we're using 3 markers, CD8, PD-1, CD11A, that we think are predictive of a T cell's engagement with the tumor. And we've been trying to study what happens to that T cell after they engage a tumor that expresses PD-L1. And right now, our favorite molecule to look at is called BIM. And we're working on the fluorescent slides to make it prettier, but we're seeing that in these PD-1 positive T cells within tumors, BIM is upregulated in these T cells. And what happens is when you look at these tumor-reactive T cells and you knock out PD-L1 from the tumors, BIM goes down in these T cells. So that's the graph in the middle. And then on the right, when we give PD-L1 antibodies to wild type, it'll restore BIM expression. But this is important because here, this is melanoma, but we have data that we're submitting for lung, and we're developing a mesothelioma cohort. These were melanoma patients before PD-1 and CTLA-4 inhibitors, but patients who had high levels of BIM, suggesting they had engagement with PD-L1, had significantly worse survival than those who did not. And then the other thing is we're seeing some patients, when we treat them with these immunotherapies, their disease will look worse and then it'll shrink, and this is called pseudoprogression. So here's a case of a patient who had pseudoprogression of this lesion, but their levels of BIM went down while they were getting, this group was treated with Pembrolizumab. BIM went down, and that's the profile we see when patients are responding to these immunotherapies. And sure enough, this patient eventually had a response to this. So we're seeing that BIM, when it changes, it's correlating with responses, and it's also helping us determine who's having pseudoprogression. So here's one patient with mesothelioma who was treated with Pembrolizumab, and by the modified pleural RECIST criteria, this was stable, but there was some shrinkage of some of the pleural disease and subcarinal lesion, and we're seeing BIM go down, which is the signal we're seeing when patients have stable disease or are responding to these therapies. So we're encouraged by this, and we have data that will hopefully be presented at ASCO this year. So we're trying to validate BIM and other biomarkers in both lung cancer and mesothelioma. These are being incorporated into some NCI-CTEP trials that are coming online in the near future. We're also using new T-cell assays to try to identify synergistic approaches. Immunotherapy is being combined with everything right now, rightfully or wrongfully, and I think we'll see some of these studies fail, but some of them will work. But we're just trying to be smarter as to how you give these drugs, in what order and what sequence, based on these dynamics of expression. So just to summarize here, a PD-L1 expression is heterogeneous and dynamic. It's currently being used as a predictive biomarker, but I hopefully have shown you data that will challenge our acceptance of just this binary presence of the marker. Right now, we have front and second line Pembrolizumab approved with 50% or greater expression. The others don't require testing, although it is recommended. So with that, I'd be happy to take questions if there's time. So.